We shall discuss now about how this biotechnology is useful in the field of medicine. We'll discuss now about insulin. So human insulin, it's a hormone naturally produced from the islets of Langerhans. These are the special patches of cells present in our pancreas. So there are two cells which are present in islets of Langerhans, alpha cells and beta cells, and we learned in detail in our lower classes. So these beta cells of the islets of Langerhans in the pancreas, they produce insulin. So this insulin's work is to control our blood sugar. So this insulin, it is formed of 51 amino acids. It is composed of 51 amino acids. So the insulin consisting of these 51 amino acids are arranged in two polypeptide chains. So these polypeptide chains are polypeptide chain A and polypeptide chain B. So out of the 51 amino acids, A polypeptide chain has 21 amino acids and the B polypeptide chain has 30 amino acids. So insulin is a hormone secreted naturally in our body and particularly in the pancreas, which has islets of Langerhans. And in the islets of Langerhans, there are two types of cells. Out of these two, beta cells of the islets of Langerhans secrete insulin. And insulin's function, we know, it is to control or lower our or maintain the blood glucose level. So chemically, this insulin is composed of 51 amino acids. And the 51 amino acids are divided into two polypeptide chains. They are present in two polypeptide chains. One is the A polypeptide chain, the other one is the B polypeptide chain. And 21 amino acids are present in the polypeptide chain A and 30 amino acids are present in polypeptide chain B. Apart from it, A and B polypeptide chains are attached by disulfide bonds. A and B chains are attached by disulfide bonds. And how insulin is controlling our blood glucose, that is, it facilitates or it makes glucose available for oxidation so that glucose is oxidized to form energy or to release energy. So this is how insulin make use of our blood glucose. So glucose is available, or we can understand it facilitates cells to take in glucose for oxidation so that glucose is broken down to release energy. So what will happen if insulin is deficient in our body? So what will happen? Naturally, when insulin is not available or not secreted in lesser quantity, naturally blood glucose level will increase. So when glucose level in the blood increases, it leads to complications and it may lead to death also if not treated. So this is about how insulin is secreted in the body and what's its chemical structure and how it works, how it functions, how it makes glucose to be oxidized so that glucose level in the blood is under control. So earlier days, that is in earlier years, insulin was isolated from animal body, that is from the pancreas of pigs and cows. So when people were in need of insulin, people who were affected by this insulin deficiency, that is who were diabetic, they needed to be injected with insulin to control their blood glucose. So in that case, so those days biotechnology was not available. So insulin was isolated from the pancreas of pigs and cows and purified, and it was used to treat diabetic patients. So what happened was the structure of the insulin of human beings and insulin of pigs and cows, it has minor differences. So due to these minor differences in the insulin structure of the insulin of humans and animals like pigs and cows, when This insulin from the animals was injected into people. It resulted in some allergic reactions in some patients, diabetic patients. So it was the technique, that is, insulin was produced by recombinant DNA later in the year 1970. So we'll discuss how this recombinant DNA technology was useful to produce insulin from human beings themselves.